Hello, my name is Aiden Udan Dunstan, and my metric number is 97977, and I am from Subgroup 7, Group 2.
Hi, my name is Asma Draman, and my metric number is 98267.
Hello, my name is Deirdre Ayen, anak Judas, with a metric number of 98698.
Hello, my name is Edna Alicia, anak Chimiam, with the metric number of 98765. The objective of the experiment were to introduce the DNS method as a reliable way to measure sugar concentration in various samples and to practice preparing cereal dilutions for accurate analysis. It also aimed to determine the absorbance of unknown glucose solutions using a spectrophotometer. Finally, the experiment applied the linear equations from the glucose standard graph prepared in Practical 2A to calculate the sugar content in selected drink products, which is tea.
Material of this experiment are shown as following. Firstly, the given sample have to be diluted with ratio of 1 to 9, where 1 part of sample diluted with 9 parts of water. Then, the following volume of sample solution and distilled water were pipetted into respective test tube according to the table shown. This was done in duplicate. After that, DNS reagent of 1 ml were added to all of the test tubes. After all of the test tubes were filled with all of the solutions, they are shaken well using a vortex mixer. Then, the test tube were placed in the boiling water bath for 5 minutes. After 5 minutes, the test tube were taken out of the water bath to cool until reaching room temperature. When cooled, 1 ml of Rochelle salt were added to all of the test tubes. Then, around 2 ml of each of the solution were transferred into the cuvette. The optical density of the colored solution was read at 575 nanometer using test tube 1 as a control. It is set as a negative control that was used to eliminate background readings.
In conclusion, this experiment used the DNS method to measure reducing sugar in a tea drink with the standard curve from Practical 2A. Results show absorbance decreased consistently with dilution, confirming that sugar concentration was proportional to absorbance. The undiluted sample had the highest glucose content at 0.984 grams per liter, while the most diluted showed much lower levels. Despite possible errors from pipetting, heating, or interference by natural compounds, the method remained effective and reliable. Overall, this experiment highlighted the value of careful technique and spectrophotometry in sugar analysis.
In the new learning section, in Practical 2B, we learn how to apply the standard curve from 2A to find the concentration of glucose in a real-world sample. By matching our sample's absorbance to the curve, we could estimate the sugar content accurately. We also practice serial dilution techniques. These help us work within the detection range of the spectrophotometer and taught us how dilution affects the concentration of reducing sugars in the sample. Another important takeaway was understanding the potential limitations of the DNS method. Since the tea drink may contain natural compounds like polyphenols, we learned that these could interfere with the absorbance reading, and that sample purity plays a big role in accuracy. Finally, these experiments reinforce the importance of good lab practices like using blanks correctly, cleaning cuvettes, and managing hazardous reagents. All of these steps help ensure our results are both reliable and safe to obtain.
Difficulty in mixing. Some samples were hard to mix evenly, especially when dealing with viscous or solid lipid samples, which might have led to incomplete solubility tests. Strong odors of solvents. The pungent smell of some solvents made it uncomfortable to work for an extended period, requiring frequent breaks or work near the fume hood. Include a control sample. Add a blank or control sample, such as pure solvent without lipid, to compare the result clearly and confirm that any observed changes are due to the presence of the lipid. As for the explained preservation parameters, this is to record additional data such as temperature, time taken for separation or mixing, or pH if it's relevant. These provide more detailed information to support the interpretation of results.
Additional suggestions for improvement, like pre-treat or filter the sample to minimize interference, because drinks like tea contain compounds as polyphenols which might affect absorbance readings. Pre-treating or filtering the sample can help remove interfering substances. This improves the accuracy of glucose quantification. And secondly, adjust dilution range for better accuracy in low concentration samples. Some diluted samples had very low absorbance, approaching the detection limit of the spectrophotometer. Introducing intermediate dilutions like ratio 1 to 5 or 1 to 15 could improve the data spread, help maintain measurements within the optimal detection range.